Hello, I'm Neda, and I'm going to briefly present our results on the studies on the antioxidant and antineoplastic activities of quaternized benzimidazole arylhydrazones. The excessive concentration of reactive oxygen species disrupts the existing homeostatic balance and hinders the antioxidant capacity of the organisms, leading to oxidative stress. The oxidative stress is considered to be a common underlying mechanism leading to damage of proteins, cellular dysfunction, and demise. This is related to serious pathological conditions such as cancer and neurodegenerative disorders. As the introduction of versatile pharmacophores in one molecule could lead to the generation of architectures able to interact with biological targets by few different mechanisms and by demonstrating synergistic activity, in our previous studies, we have successfully applied the strategy to develop benzimidazole-based cytoprotectors exhibiting antioxidant activity, which were designed as structural analogs of melatonin. Currently, we are presenting a series of benzimidazoles containing quaternary N-atom that were evaluated for their potential for combined antioxidant and antineoplastic properties. The 12 compounds were synthesized as shown on the scheme using phase transfer catalysis for obtaining of the esters. Then by a reaction with hydrazine hydride were obtained the hydrazides, which were condensed with a variety of hydroxy and methoxy substituted benzaldehydes in order to be obtained the target hydrazones. Initially, they were evaluated for their ability to inhibit the iron-induced molecular damage on biologically important model systems with lecithin as a model for lipid peroxidation and with deoxyribose for DNA damage. As it could be seen from the graphs, all the tested compounds showed antioxidant and protective effect in the model system containing lipid molecules. On the other hand, the effect on the deoxyribose varied with the structure, and based on that were differentiated the compounds with pronounced potential to initiate pro-oxidation. The highest protective activity in both model systems had the compounds containing a serine aldehyde residue, the 2-catechols 2,3-dihydroxy and 3,4-dihydroxy groups. The cytotoxicity of 3 selected compounds was evaluated in concentrations between 0.4 and 200 micromoles using a panel of tumorigenic and non-tumorigenic cells. Based on the calculated IC50 values, it could be concluded that compound 3, which is the 2,3-dihydroxy, is more toxic to non-tumorigenic than tumorigenic cell lines. On the contrary, compound 5, which is the 3,4-dihydroxy, and compound 2, which is the salicylic derivative, possess antineoplastic potential and lower cytotoxicity towards normal human keratinocytes, as it is visible from the CI values. Therefore, compound 5 and 2 could be considered as having a better potential for further pharmacological investigations on their activity and mode of actions as antineoplastic drug candidates. And thank you for your attention.